Dear students, now we are going to discuss a second type of dark reaction, that is a C4 mechanism. C3 cycle is a biosynthetic pathway. C4 is not a biosynthetic pathway. We'll see what is that C4 cycle. Is it a biosynthetic or non-biosynthetic? So the question was after the proposal of C3 cycle by Melvin Calvin, particularly Hatch and Slack scientists, Hatch and Slack raised a question. What about rest of the plants? Do all the plants are having first stable compound, phosphoglyceric acid after first carboxylation? So that was the doubt of Hatch and Slack. So scientists concerned with tracing out this mechanism of dark reaction were two members, Hatch and Slack. Hence the name Hatch and Slack pathway. And what about the plants in which they studied C4 mechanism? Scientist names Hatch and Slack. They studied this mechanism in two plants, sugarcane and maize. Sugarcane and maize. So in this experiment, what they did, they supplied 14 CO2 radioactive isotope, 14 CO2 to sugarcane and maize. And the rest of the inorganic components, H2O, and the physical one, that is light, they are normally supplied to sugarcane and maize. So here 14C was a radioactive isotope like a Melvin Kelvin. So in this experiment, what they traced out? They traced out the first stable compound after first carboxylation. First carboxylation stable product. After first carboxylation, stable product was 4 carbon compounds, particularly oxaloacetic acid, 4 carbon compounds. Malic acid also, according to your textbook. Okay, so they traced out 4 carbon organic acids, OAA, oxaloacetic acid, malic acid also. Hence the name C4 cycle. Reason they are with 4 carbon atoms. Hence the name C4 pathway. Okay, so plants with C4 cycle. It's a cyclic pathway. So plants with C4 cycle called as C4 plants. And very interesting thing is all C4 plants with both C3 and C4 cycles, both are present. Reason, C3 is only biosynthetic pathway during which C of CO2 is reduced into carbohydrate. Okay, so C3 cycle is biosynthetic pathway. Biosynthetic pathway means it is able to reduce or able to cause assimilation of carbon. Carbon is reduced into carbohydrate. Hence the name biosynthetic pathway. But what about a C4 cycle? It is a non-biosynthetic. Anabolic reaction, non-biosynthetic means by means of this there is no sugar formation. No sugar formation. But it is anabolic pathway. Anabolism, non-biosynthetic, both are anabolic pathways. Anabolic type. But there is no reduction of C of CO2 into carbohydrate. So there is no carbon reduction. Not concerned with carbon reduction. Then what is the role of a C4 cycle? C4 cycle is a mechanism, a non-biosynthetic pathway, anabolic pathway concerned with C4 cycle is concerned with the purpose of C4 cycle is to maintain CO2 concentration in the area, whatever the area, in the area where C3 cycle is being operated or taking place. So simply, what is the purpose of a C4 cycle? To supply CO2 to C3 cycle. So you can write one more statement. C4 cycle concerned with supply of CO2 to C3 pathway or C3 cycle. Reason is photorespiration. What is photorespiration? Photorespiration means light induced evolution of CO2, decarboxylation, and consumption of oxygen. Photorespiration is a waste process. So what is photorespiration? During photorespiration, instead of a reduction of carbon, carbon is oxidized into CO2. There is no formation of sugar molecules. There is no formation of carbohydrate. So photorespiration is present in C3 plants or C3 cycle. The reason is a Rubisco enzyme. Rubisco is able to perform two functions. Bifunctional, able to perform two functions, dual function. One is carboxylation. First one, carboxylation. And second one, oxygenation or oxidation. So it is able to perform either carboxylation or oxidation. If it is carboxylation, C3 cycle. If it is oxidation, photorespiration. So if it is carboxylation, CO2 is accepted by Rubisco. If it is oxidation, oxygen is accepted by Rubisco. So in C3 cycle, carboxylation is possible if the concentration of CO2 is more than the concentration of oxygen. So the catalytic activity of Rubisco depends on the catalysis. Catalytic activity of Rubisco engine depends on concentration of oxygen and CO2. Oxygen and CO2. If the concentration of CO2 is more than the concentration of oxygen, definitely C3 cycle. Otherwise, if the concentration of oxygen is more than the concentration of CO2, photorespiration. What is photorespiration? Simply oxidation of carbon into CO2 in the presence of light. So light induced evolution of CO2 and uptake of oxygen by chloroplast. In C3 plants, reason is Rubisco is able to perform two reactions, either carboxylation, oxidation, bifunctional. So the catalytic activity of Rubisco depends on concentration gradient between oxygen and CO2. If the concentration of oxygen is more, carboxylation, C3 cycle will be operated. Otherwise, if the concentration of CO2 is less than the concentration of oxygen, it leads to oxidation, decarboxylation. So according to evolution, first evolved plants, C3 plants, as C3 plants are suffering from photorespiration, which is a waste process, there is no formation of sugar molecules. To prevent a C3 cycle from falling into photorespiration, C4 cycle was evolved. So what is the purpose of C4 cycle? To maintain concentration gradient, higher concentration in the area where C3 cycle is operated to prevent C3 cycle falling into photorespiration. So by means of C4 cycle, C4 plants are able to reduce photorespiration. So photorespiration is absent in C4 plants. Reason, C4 cycle is able to maintain required concentration of CO2 in the area where C3 cycle is operated. So what is the problem of C3 cycle? Photorespiration. The role of Rubisco is either carboxylation, oxidation. If it is carboxylation, C3 cycle. If it is oxidation, photorespiration. By means of C3 cycle, carbohydrates are produced, but by means of photorespiration, there is no carbohydrate. So according to evolution, in order to provide protection against photorespiration in C3 cycle, C4 cycle was evolved, right? So that's why C4 cycle is a non-biosynthetic, just it is a supplier of CO2, able to maintain concentration, high concentration in the area where C3 cycle is operated. The purpose of C3 cycle is to maintain more concentration of CO2 to prevent photorespiration, which is a waste process. There is no formation of sugar. So to support C3 cycle, C4 cycle was evolved, right? So that's about the introduction part of C4 pathway. Next. Now, what about a specialty of C4 plants? C4 plants are tropical plants. What about C3 plants? Temperate plants. Temperate means low temperature, less temperature, 25 to 30 degrees. But what about C4 plants? Tropical plants, 30 to 40 degrees. They are able to grow at a high light intensities, high temperature. They are able to grow in sunny climatic conditions. So C4 plants are tropical plants, able to perform effective photosynthesis at high light intensity, high temperature. Right now, what about the specialty of other specialties of C4 plants? So anatomical, specialty of C4 leaves. Okay, C4 leaves. So what about specialty of C4 anatomy? So to study leaf anatomy, you have to take a vertical section, longitudinal section. So in the longitudinal section, what about the anatomical specialty present, that is cranch anatomy? This is very, very important. That cranch anatomy is present in leaves of C4 plants only. So cranch is a German word. Cranch is a German word. Cranch. It's a German word. Okay, that is nothing but wreath or garland. Simply ring-like. Wreath, garland. Garland is nothing but a ring of flowers, a chain of flowers. Okay, right? So wreath is nothing but a ring. A ring like one. What is that ring like one? We'll see. For example, this is a anatomy of a C4 leaf. And this one is a main one, that is a midvein, midrib, middle vascular bundle simply. So around the vascular bundle, several layered bundle sheath cells are present. Many layered, 2 or 3 or 4. So here, what about the arrangement? It looks like a garland, a chain of flowers or not? So ring-like arrangement. So cranch refers to ring-like arrangement of cranch refers to that reflects ring-like arrangement of bundle sheath cells. Bundle sheath cells. This is a vascular bundle. Go back to anatomy. Vascular bundle sheath. So what about the arrangement of bundle sheath cells ring-like? Many layered, not only single layered, several layers. Here I'm taking one layer. Many layers are present to show the ring-like arrangement. So what about arrangement of bundle sheath cells? Ring-like arrangement. So because of that, it is called as cranch anatomy. Now, what about characteristics of bundle sheath cells? Statement 1. So bundle sheath cells. Statement 1 with a ring-like arrangement. Ring-like arrangement. Second one, large in size. Large sized cells. Third statement, compact arrangement. Compact means no intracellular spaces. Compactly arranged. No intracellular spaces. Compactly arranged. Closely associated. No intracellular spaces. Next, fourth one, thick-walled. Cells are thick-walled. Thick-walled, impervious. Impervious to gases, gaseous exchange. Thick-walled, because of that there is no movement of gases. Next, fifth one, many agranal. This is very, very important. Agranal chloroplasts. agranal, without grana. Remaining all are present. That is one type of chloroplast. Without granum thylakoids. Rest of the parts are present. Granum absent. If grana thylakoids are absent, there is no non-cyclic photophosphorylation. Right? So this is a cranch anatomy. This is very, very important. In C3 plants, bundle sheath is present. They are also with bundle sheath. Bundle sheath present. Don't take absent. Present and without a ring-like arrangement, without cranch anatomy, without cranch-like arrangement of cells. Okay, so these characteristics are not applicable to those bundle sheaths. They're also with the bundle sheath, but arrangement anatomy is different, right? So bundle sheath cells are with the what type of chloroplast? Agranal chloroplast. What is agranal chloroplast? Normal chloroplast without granum thylakoids. Grana thylakoids are absent, remaining all are present. So if granum thylakoids are absent, there is no non-cyclic photophosphorylation. Right? So that is about cranch anatomy. This cranch anatomy is exhibited by or present in leaves only. Leaves of C4 plants. Definitely they will ask you, bundle sheath is present in C4 plants only? Wrong one. Present in C3 plants also. But the difference is anatomy. Anatomical specialty of bundle sheath cells. Both are having bundle sheath. The bundle sheath of C4 with so-called anatomy. So cranch anatomy is present in leaves of C4 plants. In C4 plants, stem is without cranch anatomy. Root is without cranch anatomy. Only leaves are with cranch anatomy. Right? So remember, what about the type of chloroplast present in bundle sheath cells? This is very, very important. Agranal is nothing. A refers to absence. Granum thylakoids are absent. That's it. Next. Now, what about mechanism of C4? Okay, so now mechanism of C4 cycle. So to complete a C4 cycle, two types of photosynthetic cells are required. What are they? First one is bundle sheath cell. Mesophyll cell, sorry. First one is mesophyll cell. Sorry. First one, mesophyll. What is the term mesophyll? Specialized parenchyma. In dicots, it is clearly differentiated into palisade parenchyma and spongy parenchyma. In the case of monocots, it is monomorphic with only one type, generally spongy parenchyma. Go back to anatomy. Dorsiventral leaf, isobilateral leaf. So C4 pathway is present in, C4 cycle is present in both dicots and monocots. Generally, most of the monocots are generally with a C4 pathway. So C4 cycle is present in both dicots and monocots. So here, how many kinds of cells are required? Two kinds of cells. In the case of C3 plants, only one kind of cell is enough. All the reactions of C3 cycle and synthesis of glucose molecule, entire dark reaction is within one kind of cell only, mesophyll cell. But here two types of cells, mesophyll, and second one is a bundle sheath cell. So first one is a mesophyll cell. Okay, so in C4 plants, mesophyll is either dimorphic or monomorphic. If it is monocot, monomorphic. If it is dicot, monomorphic. Wait. If it is dicot, wait. Here I'm giving dicot plant. If it is dicot plant, dimorphic mesophyll. What is that dimorphic mesophyll? Palisade parenchyma, spongy parenchyma. If it is monocot, generally that is a monomorphic mesophyll. What is that? A monomorphic and a dimorphic mesophyll. Dimorphic means with a palisade parenchyma and spongy parenchyma. Monomorphic means only one type, generally with a spongy parenchyma. So C4 cycle is present in both dicots and monocots. The mesophyll is either differentiated, not differentiated. If it is differentiated, dimorphic represented by palisade parenchyma and spongy parenchyma. Otherwise, monomorphic represented by spongy parenchyma. So now, what are the events within these two types of cells? So this one is a mesophyll cell, and this one is a bundle sheath cell. Okay, so these two are required for completion of a C4 cycle. So mesophyll cell. So within the mesophyll cell, the type of chloroplast. What is the type of chloroplast? This is chloroplast, dummy diagram only. This chloroplast is a granule type. Okay, so mesophyll cells, here I'm writing mesophyll cells with a granule chloroplast. And what about bundle sheath cells? They are with a granule chloroplast. The dummy diagram, remember the concept. Both are with two membranes. Okay, so this is a normal chloroplast with both grana and stroma thylakoids. And what about this one? A granule chloroplast. In 2020, they asked this question. In C4 plants, what about the types of chloroplast? Granule, A granule. So how many kinds? Two kinds. So this is a chloroplast dimorphism. This is very, very important. In the case of C3 plants, only granule chloroplast. But here, granule, A granule. The difference is all are present. Here, all are present except granum thylakoids. The difference between these two is all are present. Here, only granum is absent. Remaining all are present. With both cyclic and non-cyclic, this one is with the cyclic electron transport system only. Okay, right? So middle lamellum. This is middle lamellum. So this one is a middle lamellum. Middle lamellum is well known to you. Okay. So if they are very nearest to each other, but in the textbook it was given like that. So follow like that only. And this one is a primary wall. Primary wall. And here and there pits are present, thin areas. And continue it. So this is a primary wall. And below that one, plasma membrane. Continue it. This one is a plasma membrane. So primary wall towards inside of the middle lamella. And this one is a plasma membrane, cell membrane. Okay, so this is a thin area, pit. This area is called as pit, thin area. And continue it. This side also. Primary wall. Next, cell membrane. Okay, right? So this thin area is called as a pit. Thin area. Here I'm writing. This area is called as pit. Thin area. So between these two cells, plasma desmata, cytoplasmic bridge. Cytoplasmic bridge in both the cases. So here I'm writing plasma desmata, that is concerned with the intercellular transportation of materials, right? So mesophyll, bundle sheath. Two kinds of photosynthetic cells are required. Now, what are the reactions? So start with a C4 cycle. So this is cytoplasm of a mesophyll cell. So here I'm taking 6 molecules at a time, 6 molecules of phosphoenol pyruvic acid. But in your textbook, 6 cycles they are taking. Here I'm taking all at a time to understand sort of basics of that one. So actually, cycle 1, 2, 3, 4, 5, 6. Per cycle, 1 CO2, like that. Here I'm taking 6 at a time. So 6 molecules of PEP. So phosphoenol pyruvic acid is a 3-carbon compound. A 3-carbon compound. So accepting 6 molecules of HCO3 minus carbonate, HCO3 bicarbonate, HCO3, and 6 molecules of H2O. And enzyme required is inside only pepcase, PEP, pepcase, CASE. What is case? Carboxylase, phosphoenol pyruvic carboxylase. This enzyme is able to regulate carboxylation only. And this enzyme is present in mesophyll, cytoplasm of mesophyll cells. If they question you, pepcase, what is pepcase? Phosphoenol pyruvic. Phosphoenol pyruvic carboxylase. What is a PEP? Phosphoenol pyruvic carboxylase. So this enzyme is concerned with carboxylation of PEP with CO2. So this is the first carboxylation. So carboxylation, first carboxylation is within cytoplasm of mesophyll cells, and location of pepcase is within the cytoplasm of mesophyll cells. So what about the product? 6 molecules of oxalo. Simply I'm writing OAA, oxaloacetic acid. Oxaloacetic acid. It is a 4-carbon compound. So place of formation of a 4-carbon compound was cytoplasm of mesophyll cells. So now it is transported into stroma of a granule chloroplast. So 6 molecules of OAA is converted into 6 molecules of malic acid. So this one is a reduction reaction. During this reaction, 6 molecules of NADH oxidized to 6 molecules of NAD plus. So OAA is reduced into malic acid. Okay, right? So here OAA is either reduced, here I'm writing OAA is either reduced or transaminated. Okay, wait. So take OAA. So what about the fate of OAA? Oxaloacetic acid. So OAA now available within the stroma of granule chloroplast. What about the fate of oxaloacetic acid? it is either reduced into malic acid, like this reduction, or transaminated into aspartic acid, amino acid. So what about the fate of oxaloacetic acid? Either reduced into malic acid or transaminated into aspartic acid. If it is reduction, malic acid. If it is a transamination, go back to mineral nutrition, transamination, OAI is converted into transaminated into aspartic acid. If malic acid is produced, they are called as a malate farmers, able to produce malic acid. Or if it is aspartic acid, aspartate farmer, aspartate farmers means producers. So C4 plants are either malate producers or aspartate producers. It depends on type of the reaction. If it is reduction, malic acid. If it is a transamination, aspartic acid. So here I'm taking malate farmers. So here reaction is either reduction or transamination. Both are possible, either reduction or transamination. So here I'm taking reduction. Enzyme is a malic acid dehydrogenase. There is no space, just a dehydrogenase. Dehydrogenase. Reduction reaction. So carboxylation, reduction. Only 2 reactions completed. Now, so-called 6 molecules. It is a 4-carbon compound. 4-carbon compound. So this 4-carbon compound is transported into bundle sheath. So 6 molecules of malic acid. So now splitting or cleavage of malic acid. Splitting and cleavage. Okay, so product is 6 molecules of pyruvic acid. So here reactions. What are the reactions? 1, decarboxylation. Decarboxylation. Only 1 decarboxylation here. After that, oxidation. Here reduction, 6 NADH consumed. So oxidation means definitely from the oxidation, 6 molecules of NAD reduced into 6 molecules of NADH. Okay, so how many molecules of malic acid? 6 molecules. So in each molecule, 1 carbon is decarboxylated, right? So because of decarboxylation, what about the CO2 available? 6 CO2 available. Okay, so here enzyme required is a malic enzyme that is concerned with a decarboxylation and oxidation. So because of splitting of 6 molecules of malic acid, 6 molecules of NADH produced, and decarboxylation, 6 CO2 produced. Now 6 CO2 accepted by C3 cycle. Same C3 cycle, copy paste, and synthesis of 1 molecule of glucose. Okay, right? So what is the purpose of a C4 cycle? Supplying CO2 to C3 cycle. C3 cycle is taking place within the stroma of bundle sheath cells, right? Next, C4 cycle is not completed. Next, so this is a 3-carbon compound. Pyruvic acid is 3-carbon compound. This one is 4-carbon compound. Now 3-carbon compound is transported into mesophyll chloroplast. That is a granule 1. So 6 molecules of PA, pyruvic acid, 3-carbon compound. Okay, so now 6 molecules of pyruvic acid is phosphorylated to 6 molecules of PEP. So what about the place of formation of PEP? Within the stroma of a granule chloroplast. And here, what about the number of ATP molecules consumed? 12 molecules of ATP. This is very, very important. Here, 12 molecules. Dephosphorylated to 12 molecules of AMP, adenosine monophosphate. Okay, next. Remaining inorganic phosphates. Actually, only 6 inorganic phosphates are added, and the remaining 6 inorganic phosphates. So, actually, out of 12 ATP molecules, 12 into 2, 24. Out of 24, 6 are consumed. Remaining, so 12 into 2, 24 inorganic phosphates. Out of 24 inorganic phosphates, only 6 are added to PEP. And remaining how many? 18. So remaining 9 pyrophosphates, pyro-PP, pyrophosphates are released out. Okay, right? So here only 6 inorganic phosphates are added. So 9 pyrophosphates means each one with 2 inorganic phosphates linked by bond, double bond, O, double bond, pyrophosphate. Each pyrophosphate is with 2 inorganic phosphates, right? So this is a regeneration phase, regeneration of PEP, right? So what about the total number of reactions present in C4 cycle? Carboxylation, reduction, decarboxylation, regeneration. So like that, 1, 2, 3, 4, 5, 6 times to supply 6 CO2 to C3 cycle. So C3 cycle is operated in bundle sheath cell, and bundle sheath is with rubisco. So rubisco is present in bundle sheath only, and their C3 cycle is operated. So what is the purpose of C4 cycle? Supplying CO2. So it is able to maintain concentration of CO2, higher concentration of CO2 in the area where C3 cycle is operated, to prevent that one to fall into photorespiration. So the purpose of evolution of a C4 cycle is to prevent photorespiration. So that's why in C4 plants, there is no photorespiration, right? So PEP, simply PEP accepting CO2, product OAA. OAA is reduced into malic acid or converted into aspartic acid. Malic acid decarboxylated into pyruvic acid into here, and regeneration of PEP to run the cycle again and again. So during regeneration phase, what about the number of ATP molecules consumed? 12. And here 6 NADH consumed, 6 NADH produced. So net consumption in C4 cycle 0. But what about the number of ATP molecules stored? Next, in C3 cycle, 12 molecules of NADPH and 18 molecules of ATP consumed. So here, C4 cycle is operated in both mesophyll cell and bundle sheath cell. But what about C3 cycle? Restricted to bundle sheath only, right? So that's about a C4 pathway. Wait. Now, what about energetics of a energetics of a C4 cycle? That includes a C3 cycle also, C3 in C4 plants to produce one glucose molecule. So for example, this is a first cell, mesophyll cell. This one bundle sheath cell. Okay, so this one is a C4 cycle, dummy one. C4 cycle. So what about the number of CO2? 6 CO2 accepted. Okay, so what about the number of NADH consumed? NADH utilized, 6. Next, 6 NADH produced. And here, what about the total number of ATP consumed? 12 ATP consumed. Okay, so how many CO2 available for C3 cycle? 6 CO2. Okay, so now within a C4 cycle, what about energetics of only this one? Okay, so first NADH. NADH, how many produced? 6. 6 minus 6, 0 net consumption or net production. And the second one, ATP, how many consumed? 12 molecules. Okay, plus what about a C3 cycle? Here C3 cycle is operated. In C3 cycle, what about the energetics? First one, NADH, how many? NADPHH plus, 12 consumed. Second one, how many ATP consumed? 18. Okay, so in C4 plant, to produce one glucose molecule, what about energetics? Net consumption, net assimilatory power required. Assimilatory power required to produce one glucose molecule is 12 molecules of NADH. Actually 18 consumed, but 6 produced. Net consumption, 12 NADP, H plus, and number of ATP molecules, 12, 18, 30 ATP molecules. Okay, so when compared with a C3 cycle, how many excess ATP consumed? 30 minus 18, how many? 12 excess consumed during regeneration phase. Okay, right? So C4 cycle is not a biosynthetic pathway. What is C4 cycle? It is concerned with the maintenance of CO2 concentration in the area where C3 cycle is operated. So that's about a C4 cycle.